Beside a deep hydrothermal vent, a pale crab waves hairy claws through water loaded with invisible chemicals. The hairs are not fur, and the white coating upon them is not accidental dirt. The Yeti crab Kiwa hirsuta carries dense setae that host communities of bacteria. Related species reveal even more deliberate movements, suggesting that some Yeti crabs cultivate microbial food. Their central mystery lies in the partnership between animal behavior and bacterial metabolism. Vents release reduced chemicals that microbes can use to build organic matter without sunlight. The crab positions its setose appendages where chemical-rich fluids mix with oxygenated seawater. If you hovered nearby, repeated claw movements would look strangely like harvesting an unseen current. The behavior exposes bacteria to reactants, while keeping the crab near a hazardous boundary. Too far from venting, chemical energy declines. Too close, heat and toxic conditions intensify. Survival, therefore, depends on finding a narrow, shifting zone where microbes can grow and tissues remain functional. Claw waving, body placement, and scraping convert that geochemical boundary into nutrition. Direct observations support microbial cultivation in some Kiwa species, but details should not be generalized carelessly. The phrase bacteria-covered claws describes a living surface whose composition and function require actual analysis. This investigation follows the crab as it maintains that surface between nourishment and danger. Kiwa hirsuta was described from the Pacific-Antarctic Ridge and gave Yeti crabs their memorable common name. The animal belongs to Kiwaidae, a family of squat lobster-like decapod crustaceans. Its long chelipeds carry numerous setae, creating extensive habitat for attached microbes. Other Yeti crabs occur around vents or seeps, including Kiwa puravida and Kiwa tyleri. These species occupy different environments and display different arrangements of setae and behavior. Kiwa puravida from methane seep habitats has been observed rhythmically waving its appendages. Kiwa tyleri lives around hydrothermal vents of the East Scotia Ridge in the Southern Ocean. There, dense aggregations occupy limited warm areas amid otherwise extremely cold deep water. The species examples establish a family pattern while preserving important ecological differences. Hydrothermal vents form where seawater circulates through hot crust, reacts with rock, and returns chemically altered. Cold seeps release reduced compounds without the same intense heating process. At both systems, chemosynthetic microbes use chemical energy to fix inorganic carbon into organic material. Sulfur oxidizing or methane-associated processes may contribute, depending on habitat and microbial community. Identifying the bacteria requires microscopy and molecular analysis rather than color or location alone. The pale filaments visible on setae can include multiple microbial forms, not a single guaranteed species. When chemical flow reaches the crab, it extends setose limbs and moves them repeatedly through the water. This Q-action sequence renews contact between epibionts and dissolved reactants needed for microbial production. The adaptation expands microbial growing area beyond the crab's relatively smooth shell. Its consequence is a potentially renewable food source carried directly upon the animal. The crab can then comb or scrape material from setae using mouthparts and smaller appendages. Gut contents and stable chemical signatures can help establish whether microbial biomass is actually consumed. For Kiwa pura vida, observations and analyses strongly support epibiotic bacteria as an important food source. For every Yeti crab, however, the proportion of diet and exact microbial contribution remain separate questions. Deep vehicles document posture, collect specimens, sample fluids, and preserve microbes for genetic analysis. These four approaches connect visible motion with anatomy, chemistry, and microbial identity. Video alone shows waving, but cannot determine which compounds bacteria metabolize. Fluid samples reveal local chemistry, yet concentrations can change across centimeters and seconds. Collected setae show attached cells, but recovery may disturb spatial arrangements or favor durable microbes. Genetic sequences identify metabolic potential, while active metabolism requires additional supporting evidence. Together, these limits prevent a charming farming metaphor from becoming an unsupported certainty. One hypothesis states that waving primarily feeds bacteria by improving delivery of reduced chemicals and oxygen. It predicts stronger or differently timed movements where mixing conditions restrict microbial access. Another hypothesis proposes that movements directly gather suspended particles independent of attached bacteria. That explanation predicts similar waving even when epibiont growth is sparse or chemically inactive. A ventilatory hypothesis suggests appendage motion could also modify water exchange around the crab. These functions need not be mutually exclusive, because one movement can create several consequences. If you saw bacteria on a claw, presence alone would not prove deliberate cultivation. The stronger case combines specialized setae, repeated exposure behavior, microbial growth, and subsequent harvesting. Experiments under realistic pressure could vary flow chemistry while measuring motion and microbial activity. Yet removing crabs from their habitat may alter temperature, pressure, food, and natural orientation. Field manipulation offers realism, but risks disturbing a rare community and its unstable vent structures. Consequently, careful observation before any intervention remains essential for interpretation. The boundary itself moves as vent output pulses, currents shift, and mineral chimneys grow or collapse. Crabs sensing hotter or more concentrated flow may withdraw, pivot, or adjust appendage position. This response protects tissues while preserving microbial exposure near the mixing zone. Failure carries immediate danger because hydrothermal fluid can differ sharply from surrounding seawater. At southern ocean vents, Kiwa tyleri faces an additional thermal trap. The crab depends on vent warmth, but cannot range freely into surrounding near-freezing water. Individuals crowd within habitable bands, creating exceptionally dense local populations. Crowding increases competition for favorable surfaces and may change access to chemical flow. Females carrying developing embryos appear to move away from the hottest vent areas. That shift may protect embryos from damaging conditions, though it exposes adults to colder water. Temperature cue, relocation, developmental protection, and energetic cost form another survival sequence. Larval dispersal must somehow connect isolated vent fields, but roots and survival remain incompletely known. Vents are temporary on geological timescales, forcing populations to colonize new sites or disappear locally. Microbial gardening cannot solve isolation, yet it supports dense life where ordinary photosynthetic food is scarce. The crab also provides habitat for bacteria, increasing surface area within a chemically productive boundary. In return, microbial biomass supplies food, making the association plausibly mutualistic in nutritional contexts. Whether every epibiont benefits, the host remains uncertain because attached communities can contain varied organisms. Some microbes may be passengers, competitors, or incidental colonists rather than cultivated partners. You would need activity measurements at the scale of individual filaments to separate those roles. Predators, scavengers, and other vent animals connect Yeti crabs to the wider chemosynthetic food web. Crab feeding transfers microbial production into mobile animal tissue available to other consumers. Molting periodically sheds the exoskeleton and its attached community, forcing recolonization of new setae. After a molt, microbial propagules must attach, grow, and restore a useful coating. Early recolonization may reveal whether the host selects partners or simply exposes fresh surfaces. This cycle raises unanswered questions about where starter communities originate and how hosts influence membership. Vent mining interests add human stakes because mineral-rich deposits overlap with highly localized ecosystems. Destroying a chimney could remove habitat, chemical flow, microbial farms, and reproductive gathering space simultaneously. Recovery would depend on remaining populations, dispersal, vent persistence, and creation of suitable new surfaces. Future evidence should track identified crabs through changing flow conditions without bait. Chemical microsensors could map the reactants crossing setae during each waving cycle. Time-resolved microscopy could reveal growth, removal, and recolonization of epibionts between molts. Metabolic measurements could identify which bacterial pathways operate while still attached to living hosts. Comparisons among Kiwa species could separate inherited family traits from local adaptations to vents or seeps. Evidence for cultivation would strengthen if crabs repositioned setae to maximize measured microbial productivity. It would strengthen further if harvesting rates followed bacterial growth rather than random grooming schedules. Evidence against nutritional importance would emerge if labeled microbial carbon rarely entered crab tissues. Even then, bacteria might provide camouflage, detoxification, or another function requiring separate tests. The most difficult evidence concerns evolution, because modern usefulness does not reveal the sequence of origin. Fossils are unlikely to preserve delicate setae, microbes, and behavior together. Genomes may clarify relationships, but ancestral lifestyles will still require cautious reconstruction. At the opening vent, pale claws continue sweeping through the narrow chemical frontier. If you call them furry, the essential partnership between moving water and microbial metabolism disappears. The Yeti crab's claws support real bacterial communities, while cultivation remains strongest where behavior, chemistry, and feeding evidence converge.